So we were over here. Why doesn't it start from there? Oh. Okay. So we did tRNA, we did mRNA, we did this combination essentially where we talked about how all three of them work together to give us the protein, right? So essentially we're going to see what's actually happening as well. So just to revise, the tRNA and the mRNA joined together, the codon and the anticodon pair, like I told you the last time, why is then okay, and the pair, and then the tRNA comes in with the amino acid, and the overall thing is happening in the ribosome that is made up of the ribosomal or rRNA. Okay, so this is how it's happening. Look at this one. This one is the mRNA. These are the three codons on it. This is the ribosome, the large subunit and the small subunit. Both of these subunits come in and combine on a segment of the mRNA. And what happens is these are shown as the tRNAs. So look at this one. There's a tRNA that is coming in, and this tRNA is going to give us, so there's already a tRNA over here on this active site in the ribosome, which is basically the ribosome, as you remember, is the rRNA, right? Made up of rRNA and protein. What happens is there's an enzyme inside this large subunit that forms a bond here. And then what happens is the chain transfers here, and these are removed from this side. Like this one, look at this one, it's leaving. So as this one leaves, this moves forward over here, this entire chain and this move over here, and a new one will come in and fit over here with a new amino acid. So this happens in a sequence. You can appreciate. Okay, so this one again is showing you the sequence again from the beginning. You can see that the mRNA is being formed over here. You can appreciate that we are working on one of the strands and not both of these strands. So the strand that we are using to make the copy is known as the template strand, and it makes sense because the template is used to work, right? This entire box is the RNA polymerase, and one thing that you need to know over here is that there are two opposite words: sense and antisense, template and coding. So the template strand is the strand that we use, but obviously if this is the template strand, then this one must be opposite to the template strand, right? And it is similar to this strand. It's RNA, that's another thing, but it's much similar to this strand, right? Because this is opposite of the template strand, which means it's similar to this strand, which is why this other strand is known as a coding strand, because this is what the code exactly looks like, right? Essentially, they're both the opposites of each other. Even the coding strand and template strand are the opposite of each other, and the mRNA is similar to the coding strand because it's the opposite of template strand. We had a very detailed discussion on the DNA, mRNA, tRNA, and which one is opposite of which one. Again, because we call the coding strand the sense strand because it resembles the mRNA, so it makes sense. The antisense strand is the template strand, which is anti means the opposite, so it's opposite to the actual sense strand. And the sense in this is the sense that we make of the mRNA. Okay, that makes sense. And RNA polymerase always forms bonds from 5' end to 3' end, so you can also appreciate that here, and which means this chain is running from the 3' end to 5' end. So the 3' end to 5' end always in transcription will be the template or the antisense strand, because the other strand that is exactly similar to mRNA is the coding or the sense strand. So very easy, not very difficult. Okay, now this one shows the transcription. Once again, this is from your book. I put it in here just to show you that it's exactly the same, just a little bit different diagram. Look at this. He's working on the 3' to 5' strand. That's a fact that we know. It's the RNA polymerase, so it's a polymerase, so 5' to 3' structures are being made out of of it. And then one thing you need to know is that the RNA polymerase doesn't just blindly come in and start doing something. It has a start template. The DNA itself has a start codon and a finish codon, which means that the chain is going to start from here and it's going to be made till here. As soon as this point comes in, the RNA polymerase will detach and go away. And as soon as another RNA polymerase sees this point, it's going to attach and start working. Now, right now, you don't need to know the details of it, but when you go into A2, they'll teach you details such as lac operon and other stuff, in which there's molecules that come in and bind to, for instance, the starting codon or the start triplet. Why is it calling it a triplet? Because a codon is a triplet. It has 3 nucleotides, right? A, U, C or something, which give one amino acid. So this is why it's calling it a triplet. Okay. And then what basically happens is, if we can control transcription, how can we do that? For instance, if an enzyme comes in and blocks this path, now if the start one is blocked, the RNA polymerase can't attach and no more mRNA is made, which means no more of the protein will be made. And you know that's how the body works. For instance, if you need more insulin is a protein. What you do is you produce an enzyme that is going to come in and break down these. Okay, so my eraser here is this enzyme. The enzyme is produced, it comes in, it breaks these bonds down. And once it has broken these bonds, now the start codon is empty. And the RNA polymerase can come and bind to the start codon, and mRNA can be produced, right? Because nucleotides are free floating, they're always there. We need an enzyme to link them together and to give us something, and an enzyme to open these bonds and give us area to work in, right? And sometimes what happens is the stop triplet is blocked. So different mechanisms exist to control transcription, rate of protein production. I hope this is clear. So I hope you understand what I mean, that how we control it.
Ma'am, I have a
Yeah.
question. So can you say the RNA polymerase has two functions? One is to break the hydrogen bonds between the two strands, and one is to form the phosphodiester bonds?
No, RNA polymerase does not break the hydrogen bonds, as far as I remember. Obviously, the helicase will be present to break the hydrogen bonds.
So then the RNA polymerase just forms the phosphodiester bonds then?
Yeah.
Okay.
Or excises wrong nucleotides, breaks down weak wrong bonds between them. Okay, see, enzymes, one thing you need to know is enzymes always work in reversible reaction. If it's going forward, it's going to go backwards as well. Okay, so it's going to, so if it's forming the bonds, it can also break them if it feels that a wrong sequence has been arranged. Okay, but over here, this slide is for you to understand that in the future, you're going to right now, you need to know there's a start and stop codon or a start and stop triplet. But in the future, you'll have to know how we control transcription. So even if it comes, because AS and A level are A2 are connected for them, obviously, so a lot of students give it together. So at this point, at least you should know that if he says that we're controlling the rate of transcription or how transcription occurs, you should know that these triplets or codons have proteins and enzymes that work to block them or free them of blockade to make sure that we control this transcription. Okay, so that's what I want you to know. Any questions so far?
No, that makes sense.
Okay,
That makes
so
sense.
okay, so the next thing that you need to know is, for instance, okay, even look at this here, the gene has a promoter. Now the promoter is a region which is going to have certain triplets where the RNA polymerase will get the signal, okay, I want to attach and I want to start reading this strand, and this is my end segment. This is a stop triplet. So now the RNA polymerase will detach over here and we will get an RNA that looks like this. Now what this slide is meaning to show you is that the RNA that you get has all of the segments present in this DNA. Some of these segments may be coding or some might be non-coding. Okay, so for instance, I tell you there are introns, there are segments called introns and exons. You don't need to know the details. For instance, the red ones are non-coding, they're not important for protein synthesis. Now when this RNA forms, we have formed an RNA, right? But what goes out of the nucleus is an mRNA. It's not an RNA exactly, it's the messenger RNA. So what happens to give us this messenger RNA from this RNA? What happens is enzymes come in, what enzymes do is they cut out the non-coding parts to make sure that only the protein coding parts go ahead and non-coding parts do not, because ribosomes are not function specific about these non-coding parts. They're only function specific about coding parts. So the enzymes will remove all of these red parts, make it into splice them. Splicing, you know what splicing means? Just an English word, breaking down, cutting in the center, right? And then it gives you the final mRNA. Then the final mRNA, now, this information is not part of your A-level books, but you need to know them when you're preparing for your MCATs. So I'm going to tell you guys this over here at this point. These mRNAs, when you splice them, they're given a tail and a head and a tail. So the head and the tail protect them from degradation. Okay, just know that it's not a head and a tail like an organism, but there's obviously chemicals or extra codons attached here and here in both locations for protection of this end and this end, and scientists have appeared to name them head and tail. Okay, so that's what you need to know for now. And the details, some books might tell you, some A-level books might tell you, okay, the head is made up of guanines and tail is made up of uretidines and stuff like that, or uracils, but you don't need to know the detail now. And then eventually translation occurs and you get this amino acid chain inside the rough endoplasmic reticulum lumen. So if this is the rough endoplasmic reticulum lumen, this is where it comes in, and its post-translational modification occurs, from which it goes into the Golgi like we studied, and from the Golgi, the modified protein goes out in vesicles. So again, very easy process, just need to track it from the beginning and have a clear concept. Okay, so now we need to understand how the replication and transcription processes generally differ. So we have different enzymes, DNA polymerases and RNA polymerases. The other thing is that the nucleotides in RNA and DNA differ. In DNA, both of these strands are opened up and used. Look at this helicase, the hydrogen bondings you were talking about. I've taught this in the second lecture as well, that helicase breaks them. In the RNA formation, we have transcription, which uses only one strand. RNA polymerase is used, and the mRNA is formed, which goes outside the cell, works with the tRNA and the ribosomes or the Golgi to give us protein, whereas the DNA never leaves the nucleus. Replication occurs in the S phase. Transcription occurs in all of the phases except mitosis, because in mitosis, even my replication can't occur in mitosis, because in mitosis chromosomes are formed which are very tightly packed DNA, so it's not open for any process at all. Okay, like I told you guys that it's a closed DNA, it's very tightly bound. So will you guys be able to, will you be able to tell the difference between these two processes? I hope so, because it's relatively very different. There is not really a big reason to be confused about them. Yeah?
Understood, miss.
Okay. Okay, what do we have next? Okay, this one is again using the book to show you everything. So let's trace my pointer and go with me and follow my voice, obviously. So the tRNA with the anticodons with an amino acid attached, the tRNA will dictate, the anticodon dictates what amino acid is attached. One comes in, attaches to the codon of complementary base pairing with the codon of the mRNA. The large subunit and the small subunit combine, and as soon as the next tRNA comes in, enzymes form a bond. So hydrogen base pairing between these, and a peptide bond is formed between these two. Okay, and then what will happen, like I told you, this one will move out of the ribosome area, this one will move ahead, a new one will come in place of this with new amino acid, and the chain will continue to grow. Okay, so very easy. The first tRNA will leave, the second one will move to its place, and the third one will come into the second one's place. These two will be joined and sent ahead, and the third one will come in for the bond. Okay, just one thing you need to know over here, the start codon, like I said, on the mRNA, we had a start codon for RNA polymerase. That start codon obviously needs to be transcribed, and then it will be translated as well. that start codon is always AUG, which is for methionine. So you need to know this. And I'm saying start codon, which means it's going to be on the mRNA, right? So the opposite of it will be on the tRNA. So you don't need to remember the tRNA anticodon for methionine. All you need to do is remember AUG is for methionine. And I'm going to write it down in case you guys have missed it, okay? So AUG is the triplet codon. It starts for the basic amino acid that we need to start the chain, which is methionine. Okay, so very easy. Yeah.
Yes, Miss. Miss, how many tRNAs can bind to the ribosome at once? Is it
Okay.
2 or is it 3? 'Cause some say 2, some say 3.
So it's three. It's usually three. On a higher level, what you're going to be taught on a higher level is, which I think is the correct one and should be taught at this level as well. So what happens is that this ribosome, large and small unit, have three vacant spaces. Okay, so in these three and these vacant spaces are even named. For instance, I remember one of them is known as the P space, one of them is known as the E space, and one of them is known as the A space, right? So ape basically. What happens is in the arrival phase, the tRNA comes over here, brings the amino acid, right? And we in the first stage, for instance, this is where it's starting. The first tRNA comes to the P site. The P site is known as P site because the peptide chain forms here, and the A side is the arrival side. So what happens is the tRNA comes in on the first one, methionine comes in. This is methionine. It comes in, it waits for the arrival one to come in. The arrival one comes in glycine, for instance. Peptide bond is formed between them. Now this methionine, so what's going to happen is now this bond is going to break. As soon as complementary base pairing over here breaks, this bond breaks. This tRNA from the P site will move over here. Okay, on the exit phase, right? It's not going to wait, it's going to move over here. The methionine is going to move over here, obviously, and the G glycine is going to move over here. The bond between them has formed. This amino acid, this tRNA has moved over here. Now what happens is that this ape space is empty once again. Now what happens is another DNA comes in into the arrival space with its amino acid. The P site forms another bond. This one is kicked out of the exit phase, and this one now moves into the exit phase. This one moves into the peptide formation phase, and a new one comes in for the arrival phase. And the chain keeps growing. And you can imagine the chain has to enter the rough endoplasmic reticulum lumen. So this must be the lumen of the rough endoplasmic reticulum, right? The chain is now floating inside over here, ready to be modified. Is that clear?
Yes, Miss. So, Miss, once the peptide bond forms, it breaks the hydrogen bonds and then it reforms them at the exit phase.
No, it just breaks them, and the tRNA enters into the exit phase, which means it's lying close enough here. So imagine this, if this is the hydrogen bond, it's so simultaneous that this one is breaking and it's still trying to be there, right? But it's being kicked ahead. And what essentially happens is this entire ribosome moves along this direction. So for instance, what's happening is the tRNA isn't exactly moving, the ribosome is moving. So for instance, if this was the P phase initially, now this bond has formed. This bond has formed here, right? Now this was the P phase and this is the A phase, right? This bond has formed. Now what will happen will the ribosome will move three spaces ahead over here. Now this phase, because the ribosome has moved, this phase becomes the P phase. This phase, these three become the A phase, and this one becomes the E phase, right? The bond has formed over here and broken over here. Even if this tRNA remains attached and move during the time the bonds over here are being broken, even if it remains attached, it's still called the exit phase, right? So the ribosome has moved ahead. It has moved, it has left these three codons. Do you get what I mean?
So the EP and A phase are always changing because the ribosome's moving.
The ribosome's moving ahead. So the ribosome,
Okay.
what the ribosome is doing, it's going to read these three and it's going to read six initially, right? Which is why you're confused. The first one, the P, the A phase, right? Then the ribosome is going to move ahead from these first three to the next three, right? It's going to read these three, and then it's going to move ahead to the next three. So as soon as what happens is this phase keeps shifting. As the ribosomes moving, the P and A phases keep moving one phase, three codons ahead, right? So the automatically this one becomes the end phase, this one the exit phase, right? This one keeps becoming the exit phase. Now, even if the tRNA is attached or moves quickly, depends on the tRNA itself, but the ribosome has done its job. It has collected the amino acid. The tRNA is now free. Okay.
Okay, miss.
So is that clear? The ribosome is in fact moving ahead. If you go look at the Amoeba Sisters video, she explains it in a very nice illustration. Did you get a chance to look at it?
No, I haven't watched that one. I'll watch it today then.
Okay, yeah, make sure you watch it. I asked them very specifically to make sure they watch it. Look at this one here. So it's moving ahead and a new one is coming in. This one has moved 3 blocks ahead, right? And the old one is exiting. Look at the ribosome. It has moved ahead. It's moving ahead 3 blocks at a time. And the first one is always methionine. So is this clear? And then the last one, the first one is always methionine, and the last one is always a stop codon. And a stop codon means an empty tRNA will come in, and this ribosome will move ahead over here. But in the entry phase, an empty tRNA comes in because stop codon does not code for any tRNA. And when an empty tRNA comes in, no amino acid bonds will form and the ribosome will detach itself, and then this chain will terminate. So not the videos are amazing for it. I can send you more videos for it. But essentially, you need to understand that the ribosome moves along the chain. The first one is these, and the stop codons can be any one of these. It can be UAA, UAG, or UGA. Okay. So that's essentially translation for us. So this one is showing again the gene. So I explained this concept to them last time as well. DNA template strand gives us mRNA, which gives us codons. 3 of them form a codon, and that codon or that triplet gives us one amino acid. And there are some properties of codons that are very important to understand. So the salient features of the properties of this codon are that they exist as triplets. There are 3 bases, okay? So first, before moving on to this, I still want to talk about one thing about tRNAs. So each tRNA has an anticodon that is specific to one amino acid. And you guys know from O level that there are total 20 or 21 varying information in different books. There's 21 mostly or 20. So I say, let's say 20 amino acids in this world. There's only 20 naturally occurring ones. And one tRNA is specific for each amino acid. So how many tRNAs do we have? 20, right? Because they're specific for each amino acid. So essentially, all the nucleus needs is 20 specific tRNAs with anticodons to carry all, and many copies of those to carry all the 20 essential naturally occurring amino acids. So that's one fact that you guys need to know. Then you need to know the properties of this genetic codon. So the genetic codon is a triplet. 3 of these bases form one codon. This is important because the sequence of these will become very important in determining what codon. If it was a doublet, for instance, only A and U, then the only other option would be U and A. So having 3 of these means there can be many sequences and it's still tidy, not untidy. So it's a good combination of 3, right? Then the other fact is that it's non-overlapping. No bases are shared, which means 3 are for one amino acid, then the next 3 for another one, next 3 for another one. There is no sharing. We can't be like ACC and then start from use this CAA for another amino acid. No, this, the ribosome will read this, then the other 3, then the other 3. So there's no overlapping, there's no intervening or differences between the codons, they're continuous. Then there are two very important things to know. They're degenerate and universal. So universal is easier to explain. Universal means every organism will have the same codons. Mitochondria have variation in their starting and end codons. Some fungi have variation as well, but generally for the important amino acids, tryptophan, phenylalanine, glycine, serine, all of these generally are universal. The same three, for instance, what do I mean by universal? UGG is a codon for tryptophan. In any organism, when UGG will combine, tryptophan will come in. Nothing else can come in. UAA is a stop codon for one organism. In any organism, UAA will still be stop codon. UAG will still be stop. AUG will still be methionine and will start the protein synthesis chain. So they're universal and there's no overlapping. And the last one, they are degenerate. Now, this is a very important point. It means simply that more than one of these triplets can be used for the same amino acid or the same function. For instance, these three are different, but being used for the same function of stopping, right? That's what degenerate means, that more than one triplet can code for the same amino acid or for the same order, right? So we don't need, so essentially that means if, for instance, in my body, if I'm lacking I'm lacking a G, I do not eat enough guanine, I don't have it, will all of my stop codons stop working? No, I can still use UAA to stop myself, right? If there's a random error between A and G, will I have an error in terminating my chain? No, the stop codon for both of them is degenerate. It's the same. So a random error can still help us from getting a major change in the amino acid sequence. For instance, if I miss a stop codon here, the chain will continue for a long time, and then the protein will be larger and different than what it's supposed to be, right? So I hope that makes sense. So degenerate means that more than one triplet can code for the same amino acid. We need one triplet, but unlike tRNAs, where we are saying, okay, we need 21 tRNAs only, in codons, we don't need only 21 codons. We can have 100 codons and 100 different codons for one amino acid. We can have only 2 codons. We can have only one specific codon for one amino acid. So it's degenerate. It depends on the amino acid. And how do we know that? We know that because when we were studying, we realized that there are many combinations. But in those many combinations, most of the triplets give us similar amino acid. For example, UUA, UUG, CUU, CUC, all of these 6 are giving us leucine. Okay, all of these 4s are giving us alanine, which means that even if there is a slight difference, or if and that's so clever, even if there's a slight, as a wrong one is put in, a U is put in instead of a C, you'll still get alanine. You won't get a huge change, because then that would have led to many defects. And even if there is some is lacking, you still don't get you still get a huge range to choose from, right? So that's a very important function of having a degenerate code, a code that can have multiple variations for each amino acid. Having a degenerate code gives us the ability to have, like I told you, there were 21 amino acids, right? And 21 tRNAs, but not necessarily 21 sequence of codes. There are in fact 64 sequence of codes, and 64 sequence of codes because they are degenerate, right? Because more than one can code for one amino acid. More than one code can code for one amino acid. Okay. So for instance, this one, like I told you, proline, all of these can give proline, right? Okay, so and then the second thing you need to understand is how will you read this sequence. If I tell you I have a AUG, how will you use AUG in this table to find the right amino acid? So what you do is you read the first letter and you find it from here. I have an A. You read the second letter, you find it from up here. I have AU, and now this means that I'm going to use this row and I'm going to use this column. Then you move on to where both of these are interacting. So both of these are only interacting over here in this block, which means for the third one, I can only use the reference of this block. My third one is a G, which means I'm limited to using this line. So AUG gives us methionine, and we know that's a fact. Okay, let's do one or two more. Okay, so now I have, let's say, ACU. Okay, let's see an ACU. So for A, I have this row. For C, okay, this is C, right? Yeah, C. I can't see because there's a green thing that won't bulge. Okay, this is C. Oh, never mind. It can go down here as well. Okay, so this is the C. And then the third one, now both of them are combining over here. So this is my final box. I'm going to have to use this. And then my U is over here. So my line is this line, which means ACU comes here, and that's for the amino acid THR, which is threonine. And don't worry about learning their names. You'll get used to them, or you'll get a table with full names. So, but this is usually the table that's given. So you can just write THR amino acid as well if you don't know the full name. So let's make Raid do one, okay? Because he's here, so he needs to be punished for being here, I guess. Are you okay with doing one? Can I give you one?
Oh, okay.
Okay, so do GCG for me. What amino acid am I getting?
Alanine.
Alanine, that's correct. Lovely. See, you already know their names. Smart guy. Very nice. Okay. Now let's see what we have next. We're almost done with the topic. So for the translation part, please watch that video. I'm sure it will be much more clear. Okay, and then he's showing us the names for all of these. You guys can just screenshot this or find it on the internet. Look at the amino acids, look at the specific sequences, and then finally we are on pathologies, my favorite part of biology. So in this, the pathologies that we have are mutations. Now we already talked about cancers beforehand. So why didn't, and many people confuse mutations with cancers and correlate them, but that's not essentially true. When I was teaching you guys cancers, you guys asked me, oh, maybe there's a mutation. And I kept telling you guys that there's not necessarily a mutation. Cancers can be due to different reasons, but they can be genetic, they can be due to non-separation of chromosomes. We studied, remember all of the things we studied, and the benign and the metastatic cancer, etc. You can revise the lecture, we can go over the topic again, whatever you guys want. But essentially mutations can also lead to cancers, but mutations can also lead to differences. For instance, if somebody's eye color mutates and their child gets blue eye color, right? So that's also mutations, genetic differences or hereditary changes. Mutations can be helpful over time, like the Darwinian theory in which giraffes got longer and longer necks because the trees got longer, so the trees got taller, so there was one giraffe with a mutation with a longer neck, and they started surviving and they started reproducing more. So obviously we know DNA, whatever DNA gets passed on is more likely to produce offspring and predominate in the next generation. And this is such an interesting concept. That part of science is a lovely part because it teaches you so much about this vast world and where you stand in it, right? So mutations can essentially lead to a lot of functions. Right now, you need to know the basic of mutation. Mutation can occur at DNA, mRNA, or protein level. For instance, at a DNA level, you had something called TTT, right? And you had this triplet codon. And what happened was there was a side, there was a, so just ignore this for now. So for instance, at the DNA level, this T got replaced with an A. Now the mRNA becomes AAT, right? So this has become different from AAA, which means AAA might be for lysine, but AAT might be for threonine or glycine, and then glycine is put in at the protein level, and that changes the exact function of the protein. How does it change that? In you remember beta sickle cell anemia, what happened was valine and glutamic acid got changed. Valine was polar and glutamic acid was an acid. Sorry, valine was non-polar, glutamic acid was polar. So the entire shape of the cell changed because all of the protein was different. Similarly, if this lysine has an NH3 positive group and a glycine we know is non-polar, this one is definitely polar. So if this molecule is replaced by a non-polar one, then the entire thing changes. So that is essentially mutation. It can occur at the level of the DNA, at the level of the mRNA, at the level of the protein. Then we have three types of mutation: silent, nonsense, and missense. Silent mutation means you changed one of the amino acids, but because the code was degenerate, it coded for the same amino acid and no change took place. Silent means no change at the protein level. Okay. No silent. What does silent mean? No change at the protein level because of the degenerate code. What does nonsense mean? Nonsense means always that you replaced one amino acid by mistake or as an error, and what happened was it led to an early stop codon or a late stop codon. Okay. Nonsense means that the stop codon is introduced. Missense means a wrong amino acid is introduced. For example, if TCC was replaced during DNA replication to TGC, now the cell with TGC is going to have threonine instead of arginine placed here, and the protein will be missensed. It will be wrong. Okay. Similarly, if at the DNA level the changes haven't occurred, but for instance, I say from TCC during mRNA forming, instead of AGG, ACG was formed. Then again, the amino acid is changed. And if the amino acid is changed, for instance, we needed a basic one, but now instead we got a polar one. You can read the labels here, which means it's a missense mutation. So it can be silent, which means degenerate code, so no change, nonsense, which means a stop codon, and then missense, which means a different code, amino acid. Okay. Similarly, there are other types of point, and these are called point mutations because the one point is being changed, not the entire thing. So again, look at this silent mutation. We had UUA for leucine, and we got a C replacing the U, but it's still coding for leucine, so a silent mutation. We have a missense mutation in which we got a G replacing for the first U, and we got a totally different amino acid. And we have a nonsense mutation, which means we got a U. What happened was we got an A that replaced the center U, and instead we got a stop codon. So now the protein chain will stop earlier, okay, because the stop codon dictates the ribosome to detach, like I told you. Okay, and then there is one more which is called frame shift. What does it mean? It means the entire 3 codon frame shifts. And how does that happen? You add or remove one of the amino acid. What do I mean add or remove? What I mean is if I had UUA, and we know that there are in triplets, and I had ACG. If I add one in the center over here, what am I essentially going to do? I'm going to make one of the protein amino acid is going to be this. The other one that was initially this is now going to use this one because there are no commas. I told you it's comma-less. So now it's going to have to use these three instead of these three. This will be skipped out. These three will give a totally separate amino acid. And even if they gave the same amino acid, now this one will code with the next three, and then all of the chain ahead will be switched. the entire frame has been shifted, the entire sequence of frames. So this is called frame shift. This can also occur if you remove or delete. You don't have to just add. What if I remove this one? Now again, this is comma less, so it's going to use 1, 2, and this. And for instance, okay, wait, let me erase the eraser. Okay, so now I'm not adding anything, but what I'm doing is I am going to remove something. Okay, so we had 3 of these and 3 of these. Now I removed this one. So now my DNA or RNA is going to read U, A, and this A, right? So this sequence has again been disrupted. Now this is a different amino acid and this is a different one. So frame shift can occur when you add or delete an amino acid, and then silent, missense, and nonsense mutation is change in an existing amino acid. So are these mutations clear?
Yes, miss.
Okay, so frame shift and then point mutations where you get single changes. Okay, so again, this is from your book. You can appreciate deleted areas over here. Look how short this has become. So there are obviously, like I told you, substitution, deletion, and insertion. All three processes can occur. Deletion and insertion lead to frame shift. Substitution can give us these three criterias where we can have nonsense, missense, and silent. Okay. Insertion, deletion will give us frame shift. So I hope this is clear. You can appreciate the frame. Okay, and you can appreciate the frames are being shifted. Look at this. This one has a frame shifted to 8 bases. This one has a frame because a deletion. They're showing a deletion here. This one's removed. This one has 9 bases. This has 10 bases. And so I guess 9 bases was a normal one, and this one is deletion with 8 bases, and this one is insertion with 10 bases, and the entire frame has been shifted. These three come first, and then instead of ACT, now we have ACA and TTA, and this last C is being ignored in the protein amino acid adjustment, obviously. So the entire frame is shifted after this first amino acid. The second amino acid is different, and all the rest are also different. So the similar concept appears over here. Okay, that is it. That's our lecture. And this is highly CN, DNA, and RNA, which I feel is an amazing meme to depict both of them. Okay. Any questions?
No, ma'am.